Hello, everyone. Welcome to my talk. I will talk about my main postdoc project today, which focuses on early cell divisions in the embryo. In many organisms, both invertebrates and vertebrates, the early embryonic divisions are highly synchronized. Here is an example. This is a zebrafish embryo in the cleavage stage. There's a typical zebrafish embryo which comprises of a yolk at the vegetal pole, a blastoderm comprised of cells at the animal pole, and the last row of cells between the yolk and the blastoderm is referred to as the margin. This is the top view or the animal pole view of such an embryo at the 4-cell stage. You can see the nuclei labeled. These nuclei are visible using a pCNA reporter, which means that these cells are in the S phase. At the time point when the cell switches from S to M phase, these are the only two phases at these cleavage stages, the nuclear signal disappears. So it's a very useful marker in order to determine when the cell switches from S to M. As you can see, the initial cell divisions are highly synchronous. However, with subsequent rounds of division, you suddenly start seeing that the divisions are less synchronous. That is, here on the right, you can see that the variance of the division timing gradually increases up until the point of the 10th round of division, where it suddenly just skyrockets and you suddenly see that the divisions are highly asynchronous. So this is the process that I am interested in. I am interested in mainly two questions. One, how is synchrony brought about and lost eventually? And the second question is what the biological role of this synchrony or metasynchrony is. Importantly, in the stages when the divisions are metasynchronous, they are not spatially random. Uh, what happens is that some cell around the animal pole is the first one to divide, and this is where what we call a mitotic wave originates, which is radial in shape and it moves in all directions toward the margin. The rate at which this wave travels tells us about the extent of synchrony, and as you can see, that the synchrony and the wave speed both decrease with subsequent division rounds. The question that we started off with by asking was, how do mitotic waves arise? Uh, Long story short, I have determined that mitotic waves arise because the margin cells cycle much slower than the cells around the animal pole, and it's a linear gradient of cell cycle periods from the animal pole to the margin. More specifically, the margin has a cell cycle period that is approximately 3% longer than those at the animal pole. Now, 3% doesn't sound too much, but if we imagine, let's say, 10 cells here along a 3% cell period gradient, and if they start off oscillating with the same period, what you'll see is after a couple of oscillations, they appear to form a wave. And this is the kind of waves that we also see in the case of zebrafish embryos. What we were interested in to start with was, what is the nature of this wave? Is mitotic division timing of a cell dependent on cell-cell coupling or independent of it? Do cells uh, influence each other in this? So let's go back to the schematic again. Let's assume that here we change the period of one cell. If cells were coupled and that would determine the cell division timing, what we would expect is that the divisions would be resynchronized. On the other hand, if the cells were not coupled, the expectation would be that any introduction of period would cause their periods to drift apart further, or at least not to be reconciled eventually. So we have done several analyses to figure out which kind of mitotic waves we have, whether they are dependent on cell coupling or independent. In this regard, we have also collaborated with Edward Hanizo's lab, Irene Lee in his lab. Unfortunately, in the interest of time, I will have to skip that part. I'll just show one experiment here, whereby we have done a mosaic slowing down of some cells through injection of high amounts of histone 1 in some cells. So as you can see, these cells are in S phase, whereas all the other cells are in M phase already. The question was, would these cells resynchronize with the other cell? But as you can see from this short movie, the cells, when unsynchronized, once desynchronized, do not resynchronize with the other cells. This, in addition to other experiments, which I'm not going into right now, strongly support the idea that cell coupling has a minimal role, if any, to play in the context of synchronizing cell divisions. But this was very interesting because this led us to think, consider the problem as to how cell divisions might be patterned independent of cell-cell coupling. Because if they are not coupled, why is it, or how is it that we consistently get a gradient of cell cycle length from the animal pole to the margin? Well, literature suggests that one of the key regulators of S phase length, I earlier mentioned that it is cell cycle period gradient that causes the mitotic wave to slow down. More specifically, it is the S phase length and not the M phase length that is different between cells. One of the key regulators of S phase length is the nucleocytoplasmic ratio. Assuming that the nucleus is of the same size, a larger cell will have a lower ratio and therefore will cycle faster, and vice versa. I tested if this relationship holds true in the case of zebrafish embryos. I aspirated some cytoplasm out at the one cell stage and found that in these embryos, which have high nucleocytoplasmic ratio, the S phase lengthening occurs prematurely. And I decreased the nucleocytoplasmic ratio, on the other hand, by aspirating out a nucleus at the two cell stage, and observed that the S phase lengthening was delayed, supporting the idea that it is nucleocytoplasmic ratio that regulates the length of the S phase. However, whether this has any impact on the mitotic wave was still to be determined. Also, for this to hold true in the case of zebrafish embryo, there needs to exist a gradient of cell cycle of nucleocytoplasmic ratio. I'm sorry. So in order to test if there is a gradient of of nucleocytoplasmic ratio, I measured cell volumes, starting off with the assumption that the nuclei might be of the same size across all cells, which I have also tested, but I will not go into in this case. So when I quantified the cell sizes, binning them from animal pole to the margin, I saw that there was a tendency for cell volumes to decrease as we moved away from the animal pole. This was interesting because then it made us wonder when such an asymmetry first arises. The first time an in-out or a central periphery or an animal pole margin asymmetry arises in the embryo would be at the 8-cell stage and the 16-cell stage. So to ask if such an asymmetry in cell size already exists at this time, I compared the cell volumes of the central cells which give rise to the animal pole and the peripheral cells which later produce the margin. And I found that consistently the central cells were larger in size, and they are larger in size because of asymmetric cell divisions of their mothers. We were interested then in understanding how this comes about. One of the most fundamental concepts that has been proposed in order to explain how the cleavage furrow of a cell is positioned, therefore dictating how asymmetric or symmetric the cell division would be, is a very old rule by Oscar Hertwig. It's a 140-year-old rule. It suggests that any cell divides along its longest axis. So we started off with this simplest rule and asked if the geometry of the cell could itself be sufficient to determine when the cell divides. To that end, we identified the longest axis of segmented cells and then bisected this axis to obtain, both theoretically and through measurements, the sizes of the daughter cells. And both in theory and experiments, we see that this assumption alone is sufficient to give rise to a larger central daughter as compared to the peripheral daughter cells. This means that asymmetric cell divisions are consistently biased toward producing a larger central daughter. However, we still needed to test the idea that it is the geometry of the embryo that produces the asymmetric cell divisions and therefore the larger central daughter. To that end, we started off by considering what defines the geometry of the cells. Well, the geometry of the early blastoderm cells is defined by two key players. One is the overlying plasma membrane, and the other is the underlying blastoderm yolk interface, which together define the geometry and therefore give rise to a hemispherical embryo later on. I altered the geometry in two ways. First, I confined the embryo such that it would produce a bilobed embryo, as you can see here. I have confirmed that these bilobed embryos have a differently shaped cell size gradient, such that cells are now larger here and here, which now resemble two ectopic animal poles. And the original animal pole now lies in a valley where cells are now smaller in size. The question was, would these embryo now produce two mitotic waves instead of one? And as I play this movie, I hope you can appreciate that there are indeed two mitotic waves, one originating from each of the animal poles ectopically produced. Then I changed the geometry by altering the curvature of the blastoderm yolk interface, because it is the curvature of the cell that orients the cleavage furrow and therefore produces the asymmetric cell division. this indeed caused the cell sizes to be even more asymmetric as compared to in the control embryos. The hypothesis here was that, or the expectation here was that this would lead to a more rapid rise in the cell division timing variance. And when we compared the division timing variance, we found that this was indeed true. All these results together strongly support the idea that it is embryo geometry that patterns cell volumes and cell cycles. But why is all of this relevant? As I said, the second part of my the second question that I was interested in was what is the role of the cell cycle gradient? One of the most important events that happens at the time when when the embryo is gradually descending from metasynchrony to asynchrony is the onset of zygotic transcription. And zygotic transcription onset has been shown previously to be dependent on both cell cycle length and cell size. Therefore, I wanted to test if zygotic transcription onset is patterned in a manner dependent on these gradients. To that end, I labeled MIR430 mRNA, which is one of the first and most abundantly expressed zygotic genes. This is a live imaging. This is a movie of the mRNA that I imaged, and the cells that are actively transcribing pri-mRNA, you would see them as punctate, as you can see here. If I play the movie again, I hope you can see that the first cells to activate zygotic transcription are positioned somewhere close to the margin, and the cells around the animal pole follow later. Eventually, all cells transcribe mRNA, of course, as would be expected, zygotic transcription would occur in all cells. The same result here in a different embryo, but you can see that if we divide the embryo into three parts, the animal pole cells are the last ones to activate zygotic transcription, whereas the margin is the first one. The key question here was, if we change the geometry of the embryo, would it change the pattern in which zygotic transcription onset occurs? So we went back to the bilobed embryo where cells are larger at the ectopic animal poles and much shorter in the valley at the original animal pole. As you can see here, now we see that in fact, whereas the margin is still the first one to activate zygotic transcription, but instead of the intermediate being the second one, it is the ectopic animal, sorry, it's the original animal pole where the cells are now smaller and also cycle slower that initiates zygotic transcription first. So it is indeed embryo geometry that patterns zygotic transcription onset. Finally, the last question that we wanted to ask was, does it have any bearing in how cell fates are specified within the embryo? So we went again back to the bilobed embryo and asked if these embryos would have any defects in the germ layer fate specification. We specifically looked at CBOX EGFP, which is a mesenterodermal marker, and we found that unlike in controls where this fate was specified close to the margin, here in the valley where cells are now smaller and act like an ectopic margin, this fate was again specified. The mesenterodermal fate was also specified in a subset of the embryos. So to quickly summarize what I showed you today, we showed that asymmetric cell divisions occur in the early embryo because of its geometry causing divisions to occur in a biased manner, which produces a cell volume gradient, thereby producing a reciprocal gradient of S phase length. And either directly or via S phase length, the cell volume gradient patterns zygotic transcription onset. And this we believe could be a principle or a fundamental principle that could guide developmental robustness in order to specify where certain cell fates are specified. And the last slide of this talk would be what we think might be happening in many species. So here are four examples that we find in evolution. If we focus on some cells that are highlighted here, these cells are smaller, these cycle slower, and they all take up the mesenterodermal fate like we observe at the margin in the zebrafish embryo. So I would like to end up by raising this possibility that that this could be a causal link between the early embryo geometry and cell fate specification, and considering that geometry is such a fundamental species-specific feature, it could act as the principle on which developmental robustness is based. With this, I would like to thank CP, who has always been very supportive. Irene and Edward, who have done a lot of elegant theory, which unfortunately I could not go into today, but I would be happy to discuss if anyone is interested. The facilities and infrastructure at IST that have enabled the work, the funding agencies, and all members of the lab. And thank you all for your attention.
Great, thank you very much, Nikhil. That was a fantastic talk, and one I'd read as a chair because every question I thought of you answered on the next slide, so that was fantastic. I do have some questions while people are typing in theirs.
Mhm.
So my first is I was struck by when you showed that image of the apical view of the zebrafish embryo, the position of the nuclei was so symmetrical and so
Mhm.
characteristic within the cells. Is the position of nuclei within cells also important, or is it just the geometry of the cell overall?
Um, Sorry, are you asking if the position of the nuclei within a cell
Within a
is
cell, yeah.
important?
That's the one.
I mean, generally the nuclei are positioned more or less in the center of mass, roughly in the cell. I have not done any experiments to alter the position of the nuclei within a cell, so I would not be able to comment on it. However, I would speculate that it might have an effect because I mean, depending on the time that the cell has before it divides, if these errors can be corrected, the spindle will find the best position for itself based on the force balance. And it would position itself again in the center, roughly. However, if the cell cycle moves very fast, then these errors might not be corrected very readily. So I guess it would be to some extent linked to the length of the cell cycle.
Okay, thank you. So we have a couple of questions from Anonymous in the box. So from a cell biological standpoint, how do you think the cell volume influences division rate?
Okay, so in literature, one of the key factors that has been shown, and we have also looked at it. I could not talk about it today. Is CHEK1. CHEK1 is a cell cycle checkpoint protein, and when active, it inhibits CDK1 activity and therefore slows down the progression through cell cycle. And this is sensitive to the amount, or this is sensitive to the amount of DNA, let's say per unit cytoplasm in a cell. So it's in a way, I imagine it as a process where it's a checkpoint and it sees, okay, if the cell is large enough, it can allow the cell to proceed through further divisions, but it becomes active as soon as it realizes now the cell has very little cytoplasm to divide one more time. So it, in a way, steps in and starts to decelerate cell cycle progression.
Okay, thank you. And I guess related to your last slide, to what extent does this apply to other systems? Is geometry driven cell division observed in any other invertebrate species, such as mollusk?
Cell divisions have been like the cell division asymmetry has been linked to geometry in other studies as well. If you're asking if geometry having a long-term effect on the fate specification much later on down during the development, uh, I am not aware of any studies that have made this connection. There are some studies that have made connection, like immediate connections. Like if you look at, for instance, mice embryos, where people have talked about the positions of the cells, and therefore, in a way, the geometry that impacts what fate cells would take, for instance, the formation of the trophectoderm or yeah, so those are more immediate effects of geometry, but I am not aware of any long-term effects of early embryo geometry that have been established hours later or maybe even days later, depending on what organism we are looking at.
Okay, thank you. And we have a question from Wojciech, who says, Great talk. Do you know if the nuclear morphology changes along the animal-vegetal axis? For example,
Uh,
more stretched or compressed, which also feeds into ZGA.
At this stage, mostly not. The morphology changes after the time points that I'm interested in, where the EVL is formed and that starts to become more and more compressed, and the nuclei also start to be compressed. But at these stages, the nuclei don't seem to be compressed. I have measured nuclear volumes, and they also don't look very different. So my guess is, although I have not very systematically focused on analyzing nuclear
Yeah.
shapes, but I would guess that it is not affected.
Okay, we have another question, which is that frog seems to be different from other species in that the endoderm cells are larger, even though the cell size is larger. So how do you explain that observation?
Sorry, could you please repeat? I
Uh, the endoderm cells are larger, even though the cell size is larger. Um, I don't know if that makes sense to
in
you in
Xenopus, you
context.
mean, or where?
In Xenopus, yes.
Yeah, I've thought a lot about Xenopus because when we were writing the paper, we were also thinking very hard, especially at that time when we were writing the discussion. One of the key differences between Xenopus and, for instance, zebrafish is that the Xenopus embryos in the vegetal half have a lot of yolk, whereas in the case of so even though the total cell volume might be very large,
Mhm.
because it would also have a lot of yolk, maybe that has some impact on how cell fates are specified. That's why on that last slide, I made sure not to say that these cells are smaller, but yeah, there is also yeah, this key difference that might play a role.
And sort of related to that, could, based on the geometry and the volume of a cell, can you predict using your model when zygotic genome activation is gonna happen?
Um, I have not done it. Um, One of the key reasons is the problem is segmentation is a huge issue, accurate segmentation. But a similar thing has been done with fixed samples in Xenopus embryos, and they can accurately predict based on the nucleocytoplasmic ratio, or in that case, actually just the size of the cell, as to when zygotic genome activation can occur. So it can have predictive power, although I have not tested specifically in zebrafish.